All right, everybody. Today we're gonna be doing this egg lab, osmosis lab. We're studying the effect of different solutions on osmosis in these eggs. So essentially, we're using these eggs as a model for a cell, which is surrounded by water, either blood or interstitial fluid. And water is going to either go into or out of the cell depending on the kind of solution that it is in. So you guys are going to take some measurements as I take them and just kind of follow along here. So we learned about the terms hypertonic and hypotonic and isotonic. And so we are going to test those terms using what I have here is different jugs full of different concentrations of sugar solution. So what I basically did to make these was to put 400 grams of sugar in 1 liter of water, and then the color is just food coloring. So this one is 800 grams per liter. Okay. So what we're going to do, I have zero sugar solution, 100 gram sugar solution, 200, 400, and 800. Okay. And we're going to put these in these different cups, which I have labeled. This one is 0. And then we're going to take one of these eggs, which are in vinegar. Okay. And then take that out of the vinegar and put it in the solution. And so what we should see based on whether or not the solution is hypertonic or hypotonic, that's what you guys are going to figure out later. We're going to see the egg either shrink or grow due to osmosis, right? So the shell of the egg here will not allow water to go or the sugar to go through, but it will allow water to go through. Okay. So these are kind of squishy. I don't know if you've done this before, right? So the vinegar is just the first step in order to break down the shell. So this shell is actually broken down. Even though it looks white, it's squishy. So what we're going to do here is I'm going to put these eggs in here very gently. That's a big boy. Oh, hold on. What we need to do first is we need to measure them. So this is the data you guys are going to take. So I'm going to 0 the scale. I think we can see the scale. We're going to put that on. So egg 1 is 60.6 grams. Write that down. I will too. Egg 2 is gonna be, so egg 2 is the 100 gram one, 100 gram per liter solution, and it is 73.9 grams. Okay, put that back in the cup as well. Egg 3, this is the 200 gram one. We are going to weigh that. It's a big sucker, 80.2 grams. Egg 4, let's put this back very gently. They are kind of prone to breaking, actually. We'll get the next egg, put it on the scale. That says 69.2 grams. And then, so we got our next cup. This is the 400 gram per liter solution. We're going to that one was egg 4. The 69.2 was in the 400 gram per liter. And then we have our last one. This is 800 grams per liter solution. So very, very sugary solution. Oh, what am I doing? I'm going to put that in there. That weighs 67.5 grams. Write that one down for egg number 5. Okay, cool. I am also going to do a whole second row of those, so we have duplicates in case any of those eggs break. Okay. Now the next thing is we have to fill this cup, so this is the 0, with solution so that it is covered in water. So I'm going to measure out about 100 milliliters will do fine of solution. So here I have 100 milliliters of solution, and I'm going to put it into this cup. And as you can see, that covers our egg, okay? So 100 milliliters. And then let's go to the second one. So I'm going to go to the 100 gram per liter solution. Fill that one up. We need the 200. Let's go here. That's kind of a big guy, but yep, it works. Fills it. $400 solution. Gram per liter solution. Remember, that's a concentration. And so you'll notice that the last two are actually floating. Okay, I don't know if you can tell. And that should make sense to you because if you've ever floated in salt water in the ocean versus floating in like a lake, you'll notice that the salt helps you float quite a bit. And so just like the salt is a solute dissolved in the water of the ocean, the sugar is a solute dissolved in this water, and it is going to make the egg kind of floaty. See as it floats up right there. Okay. All right, and so here I will show you our setup. Okay, we got 0, 100, 200, 400, and 800 gram solutions. And next time we will check their new weights, and then you guys will do a little bit of analysis on this lab. Okay, thank you.